Great, thank you for the introduction, and thank you all for attending. My name is Lucas Gillenwater, and I'm excited to talk about the preceding paper that was accepted with my co-authors Lawrence Hunter, Larry Hunter, and Jim Costello. I'm a recent graduate of the University of Colorado, and I'll be presenting on GRACKLE, which is an interpretable matrix factorization approach for biomedical representation learning. And much of the research that I did during my PhD project was based on trying to better understand Down syndrome. Now, Down syndrome is a condition that is caused by the triplication of chromosome 21, also known as trisomy 21. It's the most common survivable aneuploidy in humans. Its prevalence is around 1 in 700 births in America, and people with Down syndrome are now living much longer lifespans than they have in the past. And this triplication of chromosome 21 leads to the upregulation of approximately 225 genes on that chromosome. And so this is typically about 1.5-fold increase in people with Down syndrome compared to those with typical karyotypes. But it not only affects the genes on chromosome 21, this effect is extended to the entire genome. And these effects seem to have physiological and phenotypic effects because we see a unique spectrum of co-occurring conditions that exist in people with Down syndrome. People with Down syndrome are much more likely than those with typical karyotypes to have Alzheimer's disease and also to experience that condition at an earlier age. They're more likely to have autism and epilepsy, more likely to experience pediatric hematological disorders, so several leukemias, also several autoimmune conditions and inflammatory conditions in general, as well as conditions like sleep apnea and pulmonary hypertension. Interestingly, while people with Down syndrome have an increase in all of these co-occurring conditions, they also have a decrease in some others, including less heart disease overall and less solid tumors. Now, when we look across these combinations of co-occurring conditions, we can see that there is variation existing across this heterogeneous population. For example, if we are working with a cohort that we have some clinical data, we can use that data to create a sample similarity and start to observe pockets of people who have similar clinical profiles. And if we're looking on the diagonal of the similarity matrix, where yellow is showing those with more similar, we can start to see these clusters of individuals that might correspond to these combinations of co-occurring conditions. And usually, if we include those with a disomic karyotype, as represented here in the red, they separate out because those people have less of these clinical co-occurring conditions. Now, an open question, and something that other people in this track have discussed, is do these combinations of co-occurring conditions have similar molecular underpinnings? And our hypothesis is, of course, that they do. And so if we're able to identify those molecular regulatory events that differentiate these different combinations of co-occurring conditions, this would be really useful. We could identify predictive biomarkers that we could look for early to see what someone's risk factor of some combinations of conditions might be, as well as understanding these regulatory elements might allow us to identify drug targets that would affect people experiencing these combinations of co-occurring conditions, so a drug that could affect multiple conditions simultaneously. Now, one way we might approach this is by clustering and looking at the differences, but clustering assigns individuals to a single group. And you can see just by looking at the similarity matrix that there's some off-diagonal features as well. So people have different similarities based on different combinations of co-occurring conditions. And it would be really useful to have a method that could identify both the common axes of clinical co-occurring conditions as well as some of these off the diagonal. This could be important clinically because people may come into the clinic and there may be things that are most burdensome that they would like to focus on. So it'd be useful to understand the etiology. So as a way that we might approach that, if we're looking to be able to represent these different samples and these different genes in ways that we can have samples or genes assigned to multiple groups, we turn to representation learning and specifically non-negative matrix factorization. This works well with gene expression data, which is non-negative, and it takes that gene expression matrix and decomposes it into two additive matrices that represent samples and genes and latent spaces that identify some of the prevailing axes of variations across those two different factors. So this is an unsupervised approach where the main idea is to develop these two decomposed matrices that are additive, and we minimize the reconstruction error. And then the hypothesis is that we can then interrogate those to identify these latent variables that may identify clinically important subgroups or latent variables that identify important pathways of gene regulation. And when you're looking at matrix decomposition, those latent variables are corresponding between the two matrices. So you can learn things about the latent variables in the samples if they identify some clinical subgroup to those genes that were involved in that. However, this is an unsupervised technique where you're not guaranteed to identify clinically important subgroups. And also, you may identify spuriously correlated features in your latent variables or things that aren't biologically plausible. And so to address this, we developed an algorithm called GRACKLE. And GRACKLE stands for Graph Regularized Non-Negative Matrix Factorization Across Contextual Knowledge. And GRACKLE is also a type of bird in America. And it's a non-discriminate omnivore. It's an opportunistic eater, meaning that it will eat seeds, plants, insects, even small frogs. And I think that's very similar to what this algorithm is doing in a way. It's trying to take in all the information that we have to be able to better understand the underlying biology within these groups. And so what this algorithm does is it adds two regularization terms to the loss function that will regularize the sample and gene latent spaces for the samples, a sample similarity that could be based on the clinical metadata, and for genes, a gene similarity that could be based on some gene-gene interaction network or protein-protein interaction network or gene regulatory network. And what this does is it ensures that the latent variables of samples that have a more similar clinical profile have more similar latent representations, and the same for genes. And furthermore, we can parameterize this loss function using lambda1 and lambda2 to control the amount of regularization that we perform based on the sample or gene similarity. Now, using this algorithm, we wanted to first make sure that it worked. And to do that, we performed simulation experiments. So we developed our own simulation framework. And in the simulation framework, we began with a biologically informed network model. And we identified modules of regulatory elements within that network model. And then we simulated perturbations to those modules that we could represent it and simulate gene expression data. So, for example, within this, we can identify the gray, green, and orange module, perturb those specific modules, and then do so in specific samples. And the idea is that those samples would then represent some sort of important clinical subgroup that we could use for our sample similarity. So we can do this in simulated data, separate the simulated data into training and testing samples, run our algorithm over the training data to then identify our latent representations based on the training samples and the genes. We can then fix that gene by latent variable matrix and use that with the testing simulated gene expression data to project the data into the testing sample by latent variable matrix. And so when we do that, we can then explore how well this algorithm did at aligning these latent variables between the samples and genes. And so we can do this because we know that these genes in one particular module should align with the samples that we perturbed that module in. And we could do this iteratively multiple times to ensure that we're not falling into any local optima. Furthermore, we can look at changing different factors in our simulation experiments. One thing that we did in particular is we changed the background expression noise by adding Gaussian noise. And so on the left, we have something with little noise, and then on the right, we have something with a lot more noise that's less easily separable. And the results of the simulation experiment demonstrate a couple of things. First of all, they demonstrate that regularization by the sample similarity had an extremely large effect on the alignment between our latent spaces and the results. And that's shown in the heat map in panel A, where lambda1 represents the sample similarity regularization effect, lambda2 the gene similarity, and it's much more accurate the more sample similarity regularization applied. Another thing that we observed is that overall, if we look at the average accuracy over all parameters, that adding any graph regularization helped the algorithm perform better. And we understand that because we benchmarked our algorithm against a couple of other similarly applied non-negative matrix factorization algorithms. We did it just against unsupervised non-negative matrix factorization alone. We also applied it against the graph regularized non-negative matrix factorization, where instead of applying a prior knowledge to do the regularization, you actually learn the graph from the data and then use that for the regularization. And then we also used another one that we're calling prior GNMF, and that is where we do use a prior knowledge graph for the regularization, but we don't apply the sample similarity regularization at the same time. And so overall, we observed that applying any sort of this graph regularization on average improves the model. And then if we optimize each of these algorithms for the top performing model, we see that GRACKLE starts to distinguish itself as you increase the amount of background gene expression noise in the data. So the simulations proved promising. We then applied our algorithm to some actual gene expression data. So here we applied the algorithm to breast tumor data from the Cancer Genome Atlas database. And within that database, these breast cancer tumor data has already been labeled with subtypes based on hormone receptor status that are commonly known as the PAN50 subtypes. And these are used as a gold standard because they've been pretty well benchmarked in this area for a while. And we performed 3 separate experiments where we analyzed the effect of different regulatory subtypes on the ability of the algorithm to recapitulate those hormone receptor subtypes. We looked at, just as a positive control, if we used the hormone receptor subtypes, how did it perform? And then we used subtypes that were based on other different types of omic profile, because within the TCGA database, you have multiple data sets. So we looked at how methylation clusters might affect it, as well as a combination of multiple independent omic profiles that included methylation data, protein data, long non-coding RNA data. And we saw that overall, in all these situations, we saw a similar pattern as before, that increasing sample regularization led to greater accuracy, and that GRACKLE outperformed other algorithms. And also, if we used a totally randomized set of samples, we performed worse. We validated this in another data set, independent data set, where we saw similar results. And we also tested to see whether or not the regulatory network that we used had an effect on the outcome of the algorithm, and we saw that it did. We were comparing ours between one algorithm developed between transcription factors and target genes based on message passing to string. We found that string might perform a little bit better. So then finally, we applied this to some data from individuals with Down syndrome. We worked with the Human Trisome Project that had data on over 500 transcriptomes of people with and without Down syndrome. And what we saw is that GRACKLE was able to better identify enriched subgroups of co-occurring conditions, and that within those enriched subgroups of co-occurring conditions, we would see that the genes ranking highly on the loadings were actually related to those conditions in other data sets. So an example is in B and C, we saw that latent variable 3 had higher proportions of sleep apnea, and that the second and third highest loading genes were previously related to sleep apnea in the human phenotype ontology. So in conclusion, GRACKLE is a new method for the exploration for combinations of conditions and their underlying molecular mechanisms. And you get rankings of molecules in the context of your sample metadata. And I presented this on clinical metadata, but you could apply other contexts to understand how gene regulation happens if you have metabolomic data, proteomic data, methylation data, et cetera. And this is available at this GitHub link, which you can also find in the proceedings. In some future directions that we could then further benchmark and make sure this is an extremely robust algorithm is to compare this to other topologically motivated dimension reduction algorithms like GraphPCA and some other autoencoder-based algorithms. We could also look at implementing other NMF solvers to maybe increase computational efficiency as well as reproducibility. And we want to create a package for people to use. And finally, now that we know it kind of works in these couple of toy situations, it'd be better to evaluate these latent variables by other condition differences in gene set enrichment. And so I'd like to acknowledge both of my advisors, Jim Costello and Larry Hunter, for all of their help here and their labs who provide lots of feedback, as well as members of the Human Trisome Project, and finally my funding sources, as well as all the participants with and without Down syndrome and their families for contributing data for this research. And with that, I'm happy to take any questions.
Any questions in the audience? Over there.
I have 2 questions. One, just not to make a selection for the latent space size and the definition. So I'm just wondering how you make that. But then my great question is, a lot of times when you use these maps, the feature vectors, they're not orthogonal, and it's often very, very difficult to kind of, although you can kind of cherry pick genes, it's very difficult to have interpretability there. And I'm wondering, does your regularization help that interpretability you find, or did you encounter that in many classes?
Yeah, so first of all, the number of different latent variables to choose is definitely something to consider. In these cases, for the first 2 applications, we were fixing the number of latent variables based on the modules that we perturbed. So we knew that those modules should, or we predicted that they should align with each other. And so that was the way we identified it at that point. And then we looked at within the Down syndrome data, trying to identify what number of latent variables led to the greatest number of conditions that were enriched for differences between the latent variables. So ideally, we can identify a solution that is showing greater condition enrichment, and then we could look at the corresponding matrix to see how they align. Now, for your question about interpretability, I think that's a great question to follow up on. At this point, we've looked at identifying some of, like you said, the cherry pick loadings that were higher in one versus another. And some other follow-up experiments I've done has shown that there is great gene set enrichment, in particular gene sets across these different factors, but I need to do a really proper systematic analysis to show that that differentiates from using unsupervised NMF alone to be able to answer whether or not we increase interpretability.
Thank you. You had a question as well.
Okay. I don't know if you have ever heard about the pathway avoid information in factors. It's the prior algorithm, but I think part of your framework might be relevant, or it might be reused in some way by incorporating also a pathway avoid information to improve the regularization, on the latent region of the gene. So how would you think about possibly incorporating prior information?
Yeah, I think that that's a great question. I know that there's like the plier method out there, and we could potentially compare ours against plier, as well as potentially apply another level of regularization. So we could include the graph regularization in addition to the gene set regularization and see if that helps with some of the interpretability as well.
One last question is here.
Just a couple of things. For interpretability, it's NMF, right? So all the values are positive.
Yes.
That should also have that, right?
Yes, they're all positive.
Yeah, and the other thing is because the way I see this, when I look at quicker that equation you showed, right? It looks like you could do it with interpretative update.
Yes, that's how we did it in the algorithm.
Okay, so that which is quite efficient for immunization, should be by giving up to do any projections on it. That's how you did it.
That's how we did it with the multiplicative
And the
updates.
last question was when you had, if you can put that the equation, I didn't quite get. So is that the trace that you're taking?
Yes.
So you're taking the trace of that,
Yes.
and L is
The Laplacian.
sample, and the LS is the sample, and the LG is the gene chain.
Right.
And why would that work? It's just, I mean, I'm saying it's not intuitive to me why taking the trace of how is that imposing a constant? I mean, I see it's imposing a constant, but I'm just wondering why it's imposing the right constant. What's the intuition behind it?
Well, the intuition, I think, is by, without getting too much into the matrix algebra, which I'm not sure I can explain fully, but it's by applying the traces and increasing the regularization for those that have greater similarity. So based on that graph Laplacian, you're inducing that regularization by the trace that's going to then lead to those samples or those genes having more similar values.
I think it's an extended discussion over coffee, maybe, to explain the reasoning behind it. How about that? Thank you very much, Lucas.